All right, let's talk about some of the structures that bacteria have and the functions that they provide. This is going to be a longer video because there's a lot of things to cover. We're going to start with the intracellular structures, or the things inside a cell. Then we'll talk about the cell membranes and the cell walls of bacteria and what they're made of. And finally, we'll talk about the outer structures of bacteria. And while we talk about all three of these, we'll fold in some of the medical relevance of them that will be more interesting to you as a future healthcare provider. Now, all living organisms have to have some sort of genome. And for us and prokaryotic cells, this is made out of DNA. So we call it the genomic DNA. Bacteria can also have an additional bit of DNA called a plasmid or plasmid DNA. Now, the genomic DNA is a very large molecule of DNA, and in bacteria it's always found in a circle, and it's condensed in what we call the nucleoid region of the bacteria. Remember, bacteria don't have a nucleus, but the DNA is condensed sort of in the middle of the cells, and it's represented in the red up here. Now, genomic DNA for bacteria, it's just one single chromosome. We have many chromosomes. Bacteria have one single chromosome in a circle, and they tend to carry only one copy of that chromosome per cell. Plasmids, or plasmid DNA, on the other hand, is very small. So in our little picture with these blue plasmids, you can see they range in size, or some are bigger than others. But they are small and circular, and they're what we refer to as extrachromosomal DNA because they're not part of the normal chromosome. And bacteria can have more than one plasmid. That is, they can have multiple copies of the same plasmid, like these two that are the same size here, or they can have multiple types of plasmids, so this small one and this large one. Next up, we have ribosomes. Ribosomes are what the cells use to make proteins, and they are these multi-subunit proteins themselves that have what we call a small subunit and a large subunit. Now, hopefully you remember that the DNA of an organism contains the genes that are then made into a messenger RNA, and it's this messenger RNA that directs the ribosomes how to make an individual protein. Now, we're not going to go into all of these different subunits or how ribosomes work, but I want you to know a couple of things about them that are important for this class. First of all, ribosomes come in different sizes. So prokaryotic ribosomes are what we refer to as 70S ribosomes. S stands for Svedberg, which just has to do with their size overall. Whereas eukaryotic ribosomes, like our ribosomes, are what we refer to as 80S or 80 Svedbergs. That just means that they are different in sizes, where eukaryotic ribosomes are a little bit different, but they work exactly the same between prokaryotes and eukaryotes. And one of the really interesting things about these size differences means that ribosomes, bacterial ribosomes, are a really good target for antibiotics because they're a different size and a slightly different structure from ours. We can inhibit bacterial ribosomes without hurting our own cells. Next up, we have membranes. All living organisms have a plasma membrane. It keeps things inside the cell and it keeps the external environment outside of the cell. And membranes are made up of what we call a phospholipid bilayer. That means it's a layer of two phospholipids that have a hydrophilic head group, shown in red here. So that's the part that interacts with water, either in the cytoplasm or the external environment. And then these two hydrophobic groups on the inside. But cell membranes also have a number of other components embedded within them, like proteins. And these proteins can have jobs like being a channel through which nutrients or water or other things can pass in or out of the cell. They can have some structural proteins that have some sort of job. They can have proteins that interact with the skeleton of the cell and help the cell maintain its shape. Another really critical component of cell membranes is cholesterol, which we know of more from like getting your LDL and HDL checked. But in cell membranes, cholesterol is really important because it maintains its integrity and keeps the membrane from melting when it gets too hot. So let's just write that out over here. And the final thing I want to say about membranes is that they are what we call a fluid mosaic. Because they are essentially lipids, like an oil, they can move, they can bend, they can flex, and they can move in a sort of undulating pattern if needed, but also things can move through them. So a protein that's found within that membrane can move laterally through the membrane to another spot. Okay, let's talk about the cell wall. This is one of the more complicated structures of bacteria, and we're going to kind of just do an overview of the components that make up the cell wall. So the cell wall is made up of what we call peptidoglycan, and this is an alternating pattern of this green circle and this blue circle. Now this green circle is called N-acetylglucosamine, which gets shortened, as lots of things do in microbiology, to NAG for N-acetylglucosamine. And then this blue circle here, which is N-acetylmuramic acid, which gets shortened to, as you may have already guessed, NAM. So these long alternating chains of NAG and NAM get linked together by the bacteria, and then these little amino acid cross bridges are made to hold the layers of NAG and NAM together. Now, I like to describe how this structure is held together by thinking of a raft. So let me draw you a picture really quick. Okay, so there was my super fast drawing of some layers of NAG and NAM in blue and green, just like our diagram over on the right. So now think of each of these layers as a board or a log that you are using to make a raft to escape, a desert island somewhere that you've miraculously or mysteriously been stuck on. And if you just lay these boards side by side and push them into the water, they're all going to go everywhere, right? They're not going to stick together. So you need something to hold them together. And that something on a desert island to hold your logs together would be some sort of rope or vine. And in the context of peptidoglycan, that rope or that vine would be these amino acid crosslinks. So they come together and hold our whole big raft in place so that it stays together and doesn't just fall apart as you're trying to escape the desert island. Or rather, so that the layers of peptidoglycan don't just disperse and fall apart, but instead stay as a flat layer and continue to protect the bacterial cell. So bacteria themselves come in two really important groups, what we call gram-positive and gram-negative bacteria. And this just refers to their cell envelope. And really, envelope is just a fancy way of saying the arrangement of the cell wall and the cell membranes that these bacteria have. So let's start by looking at the structure of this simpler type of cell, which is the gram-positive. So in this diagram, we have the cytoplasm here, which remember is on the inside of the cell. We have what's called the periplasmic space, which simply means the space in between the cell wall and the cell membrane. And then on the external side of the cell membrane is the peptidoglycan cell wall. And in our gram-positive bacteria, this means that they have multiple layers of peptidoglycan. So they have a very thick cell wall. Positive cells also have what's called teichoic acid embedded into their cell wall, and this is something that's only found in gram-positive cells, and it can actually be used as a diagnostic to determine whether a person is infected with a gram-positive or a gram-negative bacteria. Okay, now let's look at our gram-negative cells, which are slightly more complicated in their cell envelope. So again, down here is the cytoplasm, or the inside of the cell, and our gram-negative bacteria have what we call an inner membrane. Now this is the same thing. It's made up the same as the plasma membrane from our gram-positive cells, but it's called the inner membrane because these bacteria also have an outer membrane. Now between those inner and outer membranes is the peptidoglycan cell wall, which for gram-negative bacteria is very thin, and it's made up of a single layer of peptidoglycan, not multiple layers, just one single layer. Gram-negatives do also have a periplasmic space. Again, that's the space between the inner membrane and the cell wall. And like gram-positive bacteria, they do have something on that outer membrane that only appears in our gram-negative cells. And that's this really long word, lipopolysaccharide, which we shorten in speaking to LPS. Lipopolysaccharide is really important medically, and it's made up of 3 parts. So the L stands for lipid, the P stands for poly, and the S stands for saccharides. So the polysaccharides are these red and blue shapes that you see here, and they're just a bunch of sugars that are linked together, attached to that lipid A portion. Now, the parts of the lipid A and the polysaccharides can vary from bacterial species to species, but the lipid A portion is the most important medically because it is actually the toxic portion. It causes those fever and chills, those aches and pains that you may feel when you have a bacterial infection. And that's just your immune system responding to this toxic bit of lipid A. Now, sometimes people notice that when they start taking an antibiotic, they feel worse for a little bit. So those symptoms of fevers, chills, aches, and pains will actually get worse a day or two after starting the antibiotic. And that's because the LPS is being released from those gram-negative bacteria as they die. And when it's released, it causes an increase in inflammation. But this is only temporary. And so we encourage patients to continue taking those antibiotics, even though they may feel worse for a while. And then, in fact, they will feel better if they're just patient and wait a couple more days. Okay, it feels like we've already covered a lot of things that bacteria have outside the cell, but there are 3 other big structures that bacteria can use to survive, to cause disease, and to belong in their environment. 2 of these things are kind of similar, and they're called the capsule and the slime layer. Now, the capsule sometimes also gets called a capsid. I use the term capsule because capsids can also refer to viruses, and I don't want us to get confused, so I will always call it the capsule. Now, both the capsule and the slime layer are made up of carbohydrates or sugars, and they function to protect the cell from the external environment. So let's look first at the capsule. In these little diagrams, this sort of brown part in the middle is the bacteria itself, and the circle around it would be the capsule of the bacteria. So if you look below, what you're seeing in these little black dots down here, those black dots would be the bacteria, and that clear area around it would be the capsule. Now, the capsule is very, very thick, so all of that clear area would be the carbohydrates that make up that capsule. And just to the left over here is a microscope image of some bacteria that have been stained using a special stain called the capsule stain. And again, these 2 little dots in the middle are the bacteria, and the large halo you can see around it is the entire capsule. And up here are 2 bacteria that do not have a capsule. So you can kind of see the capsule itself is bigger or wider than the individual bacteria. So we could describe the capsule as being compact, as being closely organized, and as being fairly dry. Now, slime layers are made up of carbohydrates like capsules are, but they look a lot different, and you can probably guess how they're going to look just by thinking about the name. Here is our bacteria in this sort of brownish color, and the line around it indicates the slime layer. Looking at the little diagram below, you can see that there's not really any sort of halo around these cells. So using that special capsule stain, you don't really see anything. However, when you grow bacteria that make a slime layer on a plate, on a petri dish, you can see that the growth itself is slimy, which is where the name comes from. It is very sticky, it can sometimes appear snotty, and it's also very shiny as well. So we would describe the slime layer as loose because there's no compactness to those carbohydrates. Compared to the capsule, it is not well organized at all. And as you can imagine, this one is pretty wet. Now, both the slime layer and the capsule we think of as being around individual cells or cells of the same species within growth. But the final thing on our little diagram here is the biofilm. So let's switch slides and take a look at that. In this part of the diagram, you can see that there are multiple cells encased together in some sort of substance. And that layer of stuff is made up again of carbohydrates, like our capsule and slime layer, But it can also have polypeptides, which are amino acids and short proteins in there as well. And together, this layer is called the matrix. Looking at the picture below, you can see that this structure does not look anything like what we have around our little cells over here. It doesn't look like the slimy growth that we see on a petri dish, but it is much more three-dimensional and thicker and has just more texture to it than either the capsule or the slime layer. Biofilms don't just form overnight. They take a series of steps in order for a biofilm to begin and then mature into something like you would see in that last image. And the first stage of this is the initial attachment, where a few cells attach to a surface. Once those bacteria cells have attached, they can start to grow in the second step, which is called permanent attachment. So we can see we've gone from a very few attached cells to now maybe some cells that we can see kind of like a colony on an agar plate. Once the bacteria have managed to permanently stick to our surface, whatever it is, they undergo the first stage of maturation, which is very cleverly called maturation 1. And in this stage, they're beginning to lay down some of those polypeptides and polysaccharides that are going to make up that matrix. The next part of this process, step 4, is called maturation 2. And in this step, our biofilm is becoming more complex. So we can see that it's grown a lot. And within this biofilm itself, you can see these little sort of pockets. These pockets allow water and nutrients to move through the biofilm so that the cells down inside don't lose access to those resources. And then eventually, these biofilms will get big enough that they hit our 5th step, which is dispersion. At this final stage, some of those cells are going to leave the matrix, start swimming out into the environment, and find a new place to colonize. One of the things that I find most fascinating about biofilms is that in nature, they are generally made up of multiple different species. So it's not one type of bacteria that makes up this biofilm, but it can be 5, 6 different species of bacteria in this one area. It's also how most bacteria are found naturally. So in the natural environment, attached to rocks or other surfaces, they tend to grow as biofilms. And they are best at attaching to what we refer to as abiotic surfaces, which means surfaces that are made of non-living substances, like, or even your teeth. On those mornings that you wake up and your teeth feel extra fuzzy, that's biofilms of bacteria that have grown up overnight. So aside from your teeth, biofilms of bacteria can form inside the body and can cause a lot of trouble. One of the most common places that biofilms form are on any sort of medical implant or medical device, in particular, pacemakers. Now, because bacteria and biofilms love to form on those abiotic or non-living surfaces, they'll grow on the implant, and once they hit that final stage, they'll break free and get into the bloodstream, causing what's referred to as sepsis. And for a pacemaker, this can be particularly bad because, right, the pacemaker is implanted here really close to the heart. So once that bacteria breaks free, it can get right into the heart itself and be pumped throughout the body, infecting multiple organs and causing all sorts of disruption in your patient. The other implant of large concern is the use of catheters, and in particular, the use of catheters in people who are in long-term care. And that's why you'll hear about catheters needing to be removed and replaced fairly often to prevent bacteria from growing on them, causing bladder infections, kidney infections, or becoming systemic as it gets throughout the entire body. Continuing on with these external things, we have fimbriae, which are these short hair-like bristles that you can see covering the outside of this bacterial cell. Fimbriae are used for attachment, and usually that's to attachment of body surfaces, so they can use fimbriae to attach to the surface of your lungs or your colon, but also to abiotic surfaces like pacemakers, or in the environment, things like tables or the inside of your sink. Pili are also appendages that come from the outside of the cell, and they can have many pili as well. These are longer than fimbriae, as you can tell just from the pictures here, and they can also be used for attachment to a variety of surfaces. But one of the important things that pili are used for is conjugation, which is a way that bacteria can share DNA from one cell to another, and even between species. And one of the important things about conjugation is that it's actually a way in which antibiotic resistance genes get shared among bacteria. Just a few things left, friends. Next up, we have flagella. You've probably all heard of flagella before. They're these long, thin appendages that come off of our bacteria, and they're used for locomotion. This type of locomotion is generally called swimming of a bacteria, and it's carried out by movement of these flagella. The flagella themselves are thin, hollow tubes, and they move like a propeller on a boat. So they turn either in a counterclockwise or a clockwise direction, and the direction at which these flagellar motors are turning determines whether the bacteria is going to run in a straight line or tumble randomly in their liquid environment. And we'll talk more about this in just a second. Now, not all bacteria have flagella, but those that do can have a variety of numbers or locations of flagella on the cellular body. When the bacteria has a single flagella coming off of the end of it, and most bacteria that have flagella tend to be bacillus-shaped, this is called monotrichous. Mono meaning one, solar flagella. Amphitrichous is where we have a single flagella coming off each end of the bacteria, kind of like being ambidextrous and using both hands equally well. Lophotrichous, lopho means tuft, so it's like having a little tuft of flagella coming off one end of the bacterial body. And then peritrichous is where we have flagella coming from all over that cell body, from each end, from the sides, from anywhere that you can find a flagella. Now, when flagella are used to move through a liquid or a semi-liquid environment, like I said, bacteria can move in either a run pattern or a tumble pattern. The run pattern is when our bacteria are rotating those flagella counterclockwise, and they're going to move in a straight line. The bacteria run in a straight line toward an attractant, like a nutrient that they need, and they run in a straight line away from something that would be bad for them, like a toxin or a bad environment. And when they lose the attractant, they turn into what's called our tumble. This is when the flagella rotate clockwise, and the bacteria are essentially just kind of spinning in place until they find the attractant again, in which case the flagella, which during the tumble in our peritrichous flagella are found all over the bacteria, they will come together and kind of coalesce at the back of the bacteria so that it can then run again toward the attractant so that it can find whatever nutrient it needs or whatever that attractant happens to be. Now, they can switch really quickly back and forth between the run and tumble and switching between counterclockwise and clockwise flagellar rotation. And I'll link a couple of videos on World Class that show this flagellar movement and the coming apart of these flagella and then the rebundling of them really nicely because it's fun to watch under a microscope. The final thing we're going to talk about is endospores. And I guess this technically is not an external structure because it's a whole other type of way in which bacteria can live. And if you remember, the endospore forms are the dormant forms of bacteria. So the vegetative cells are the dividing, thriving cells, and endospores are more of this dormant, not metabolically active form. And the function of an endospore is really just to allow the bacteria to survive when the environment has become hostile. And an important thing to remember is that only gram-positive bacteria can form endospores. Gram-negative bacteria are unable to do so. So let's take a little overview of this process. For cells that can carry out endospore formation, they're going to start by replicating their DNA. And then an extra membrane forms around one of these packages of DNA. It continues to make more and more membranes that surround that DNA, but notice that it's just this really small area of the cell. There's not a lot of cytoplasm, there's nothing else really in there. And after adding multiple layers and forming what's called a cortex, That spore is going to get released into the environment where it can lay dormant for years and years and years. Some spores have been found that are approximately 20 million years old from the guts of insects that were encased in amber. And when put in the right conditions, they were able to start growing in the lab. Isn't that wild? We can look at endospores using an electron microscope, which is the picture here in B. This black in the middle, that's the really compact DNA, and all of these other layers are the cortex of that endospore. So it's very hardy, it's very sturdy, and that DNA is well protected. We can also do a particular type of stain in the lab called an endospore stain. And this shows us the difference in which cells are vegetative or dividing and which are the endospores. And in figure C here, the red cells are the vegetative ones, so they're the ones that are actually dividing. And then if you can see the tiny little green dots in there, I'm trying not to cover them up with my pointer, but just to sort of highlight them, those are the endospores themselves. So really the challenge of endospores is that they're not easy to kill because they're not dividing and because they're in this dormant form. Even really high heat and really strong chemicals can't kill them. So medically, they can cause a lot of problems. Now, even if you aren't aware that they form endospores, you've definitely heard of some diseases that are caused by endospore-forming bacteria. The most famous, or more like infamous, genus of bacteria is that of Clostridium. You've heard of quite a bit of these because you've heard of the diseases tetanus, you've probably heard of botulism, you probably learned about gas gangrene in history when you were learning about different wars, and because you're interested in becoming a nurse, I'm going to guess that you've heard of Clostridium difficile, better known as C. diff. The image here is showing us just a general bacteria from the genus Clostridium, bacillus-shaped with lots and lots of peritrichous flagella. And you probably know that C. diff is a really big problem in hospitals because it's able to spread from patient to patient in ICU really quickly. And we'll talk about that later on in the term when we're talking about infectious diseases. Now, tetanus used to cause a much bigger problem back in, the 1950s, 1960s. But once the vaccine for tetanus was introduced, we can see that most cases in the United States have really leveled off. But one of the scariest endospores, at least from the news media, is that of anthrax. The bacteria that causes anthrax, like most endospore formers, are found in the soil, and they can get eaten by ruminant animals. And once that endospore gets into a favorable condition, like the gut of an animal, they undergo what's called germination and begin to replicate and can cause disease. All right, let's do this really rapid review. Bacteria have to have DNA. This can be genomic DNA, or it can be plasmids. They have to have genomic DNA. They may or may not have plasmids. The cell envelope, right? We have a gram-positive and we have gram-negative cells. And that depends on the makeup of the cell membranes, which are lipid bilayers, and the cell wall, which is made up of peptidoglycan, or PTG. Outside of the cell, we can have things like the capsule, slime layer, and biofilms. Bacteria can also have pili, fimbri, which is another word I can never spell correctly, fimbri, which are used for attachment, and flagella, which can be used for locomotion. And of course, we can't forget those endospore-forming bacteria and the role that they can have in human health. Okay, y'all, that's it. Next time we'll talk about fungi and algae and some of their structures and things that they have that are different from bacteria. Have a great day.